In this 2-minute webinar, we'll see how to extract the sequence of a gene, a protein, or of a chromosomal region. Okay, the first thing we're going to do is to find our gene of interest, which means we have to make sure we're in the right database. So I'm going to change our current database to a Mycobacterium tuberculosis database. One simple way to navigate through all the organisms in Biopsych is to simply type in a strain name or an abbreviation of a strain name. And then we'll click on the full name of the organism to make that our current genome. Let's then enter a protein substring name of interest. Here are all our search results. We get a pathway result. We get a number of protein results. Let's click on this one. Here's our gene of interest. There are several ways to get its sequence. We can retrieve its amino acid sequence here and cut and paste this from our web browser to a file of interest. We can also click on Get Nucleotide Sequence to retrieve the nucleotide sequence of interest. Initially, what we see is the nucleotide sequence of the coding region. So we see the ATG and the stop codon, but we can actually use this same tool to select the sequence of an arbitrary genome region on this replicon. This small window came up to the side. I'm going to drag it in here. And what this small window lets us do is to select this arbitrary genome region. So for example, let's say we want to select the nucleotide sequence for this region plus another 100 bases upstream. I can simply add that 100 to the upstream coordinate and click Show Sequence, and there we have it. I can also ask for, say, 200 bases downstream, click Show Sequence. So we now have more sequence downstream. I can also use this control here to say add another 10 bases upstream and another 10 bases downstream. More generally, using this tool, we can select any region of the entire genome. And we can also ask for the complement of the coding strand.